Bismillahirrahmanirrahim. Assalamu alaikum. Welcome to my channel Biology Learning Options. If you are new to the channel, so please subscribe and press the bell icon button for new or latest updates. So today I am here in front of you with a new topic that is basically a research topic that was conducted in order to check the impact of the water stress on the morphological parameters and the phytochemical composition of Ocimum Basilicum basil plant. So here showing this content that will be discussed in this video, that are introduction, literature review, aims of objectives and the methodology, results and the discussion, conclusion, further aspects and then the references. So in short or brief, if I talk about the basic purpose for this conduction of the research, that was associated with the investigated the effects of the different water stress conditions on the morphology, secondary metabolites and the aroma of the basil plants. So the study used basically a control experiment to grow and multiple groups of the Ocimum Basilicum plants under different water stress conditions and then collected the data on the morphology, secondary metabolites and aroma of the Ocimum Basilicum plants at different stages of growth. So the phytochemical analysis of this Ocimum Basilicum plant in methanol extract showed the presence of tannins, saponin and steroids, but the absence of the flavonoids and alkaloids in all the groups. So the research found that the day 2 group had longer and wider leaves and more leaves, while the control group has highest powder leaves weight, and the day 2 group had the strongest aroma. So this study basically suggests that farmers and plant growers can adjust the water stress conditions to optimize the aroma of the Ocimum Basilicum plants, which can lead to the production of higher quality herbs. So these results of basically this research study shows that there are potentially impact the field of the agriculture and the food science by providing insight into the impact of water stress on the growth and chemical composition of the Ocimum Basilicum plants and contributing to the development of new cultivation strategies for the Ocimum Basilicum plants. So basically this plant is basically included in the medicinal plants that are herbal medical plants that can be used for the preparation of drugs for disease prevention from plants since prehistoric times, and people can prevent certain type of diseases by preparing the drugs from these medicinal plants. According to WHO, 80% of the world's population depends mainly on the traditional medicines, which are also called as the herbal medicines. And if we talk about the study of these medicinal plants, that is called basically herbalism. And the term medicinal plants is used for a wide plant species in herbalism, in which some have medicinal activities to cure many diseases because of their therapeutic values. So throughout centuries, humans relied on nature to cater for their basic needs, in addition from nutritional requirements to the healthcare system. So here is showing the brief introduction of the Ocimum Basilicum plant. It is an annual aromatic to perennial sweet smelling herb. Basically it belongs to mint family, that is Lamiaceae family, and range from 30 to 160 species and have large morphological groups. It means it is widely distributed. And the Ocimum originates from a Greek word that is Ocimum, which means aromatic herb, and Basilicum originates from word Basilicus, which means king. So that is why the reason Ocimum Basilicum is also known as the king of herbs. It is also widely grown in Pakistan, South Africa, India and Russia. Moreover, it has different names depending on the location. It is known as sweet basil and common basil in English. In French it is called as the basilica, and it is called as the rehan in Arabic, and the holy basil and nyazbo, that is the common name for it in Pakistan. If we talk about its propagation, that is basically done generally from the seeds. These are herbaceous and shrubby plants. And if I talk about its optimal growth condition, that is basically warm temperature in full sun, but there is tolerance in light shade also has been seen. So hot season is preferred by Ocimum Basilicum plant, so it grows best in long full sun conditions. If we talk about its base temperature, that is basically 10.9 degrees C. However, the optimum growth and germination temperature for Ocimum Basilicum plant is 20 to 25 degrees C, which increases the presence of the volatile oil contents in its leaves. And due to tender plant tissues, it cannot tolerate drought conditions. Ocimum Basilicum has high water requirements with fertile soil and high content of organic matter. So it is one of the aromatic and medicinal plant that has promising beneficial effects in the healthcare system. If I talk about its uses, it can be used as a food, as its raw leaves that are fresh and properly clean can be used as a vegetables, can making in the pesto sauce, or can be added into tea or soup and can be used as a green tea. If we talk about its other uses, that are the allopathic medicines and the manufacture of the cosmetics. So here are showing some of the parts of the Ocimum Basilicum plants, that are flowers, leaves and the seeds that can be used in making medicines from these plants. If we talk about its morphology, so here are showing the parts of the plants Ocimum Basilicum and the physical appearance. So if we talk about leaves, its leaves are rounded and all in shape. They are slightly cubed and the green color to dark purple, and they can grow up to 4 inches long, that is basically 10 centimeter. Next, if we talk about roots, its roots are tap roots that are thicker and grows relatively straight down, and they vary in color according to the type of the cultivar that is fertilized. So sweet basil has green stems. If we talk about the flower, these are basically small and white in color and arranged along a spike that grows from the tip of the stem. Flower falls off, and then there are the dry structures that appear, which basically contains a seed. And if we talk about its seeds, that are small, dark and the edible. Its stems are basically square and slightly hairy stems with leaves that grow on the opposite sides and be 12 to 50 inches high, basically 30.5 to 125 centimeters. There are also nodes that are present in the basil plants. Nodes are the basically point that where the leaves are attached to a plant. And then there is internode. Internode is basically a point that links two nodes of a plant, and it is 1.6 centimeter in length in basil plant. Then there was water stress that was given this plant. It can be of two types. It can be a drought condition, that is basically a low amount of water, and it can be an overwatering, that is high amount of water. So morphological and physiological parameters of the Ocimum Basilicum plants, mainly the photosynthetic capacity, adversely affected by the water stress. If there is a prolonged water stress production and the growth of a plant can fully or partly diminished, means it can be fully deflate. So the plant have evolved to a variety of incoming environmental stresses such as physiological and biochemical adaptations by alter cellular metabolism and generating defense mechanisms. So it proves that water is one of the most limiting factors in growth regulation and development. There may be many physiological changes. Number of stems, number of leaves, number of internodes can be different due to the water stress that is applied to the plants, and there should be reduced productivity and dry environments yield, and even there is wilting, so there can be death of the plant. Medicinal importance of the Ocimum roots, that is basically anti-malarial, anti-diabetic and antimicrobial. Ocimum seeds have been shown antioxidant, anti-ulcer and anti-diarrheal properties, and the Ocimum leaves have therapeutic indications against anorexia, which is a condition that is the dryness of the skin, dermatitis, that is the inflammation of the skin, gastrointestinal spasm, laryngitis and the gastritis, intestinal parasites, cough, antibacterial, antimicrobial and the antioxidant properties. There are a number of secondary metabolites that are present in the plants of Ocimum Basilicum. Secondary metabolites are basically the volatile compounds. We can say that these are the bioactive compounds that are present in the plants. These bioactive compounds naturally produced by the plants. These are also called as the phytochemicals. Basically, these phytochemicals provide the plant-derived medicines. And these metabolites present in a plant depends on the environmental factors and control treatments such as water stress that can alter these compounds, synthesizing the natural products. And these compounds accounted for antimicrobial, anti-inflammatory, antioxidant or other biological activities. So the aims and objectives of this study was to determine the impact of different levels of water stress on basil's growth and development, to identify the secondary metabolites in basil, to compare the morphological features of all groups to check the effect of water stress. So the methodology that was performed depends on the various steps. Number one is the plant cultivation and its growth, and then there was the extract preparation, and then performing the phytochemical analysis. So the procedure for the extract preparation basically includes the collection of the leaves, and there was drying process, and then grinding of the leaves was done for mixing with methanol extract and then shaking through a rotatory shaker. So drying was done basically, then the leaves were collected and washed with distilled water and shade dried to prevent loss of the secondary metabolites. And the grinding of the leaves from all the groups were grinded using a mortar and pestle to make a fine powder and then sorted in airtight tubes for further use. In mixing with solvent step, 0.25 gram powder from each group was mixed in 10 ml of methanol Then the extract was held in rotatory shaker for 24 hours at temperature 43, then stored in the refrigerator. Then how the measurement of the morphological features was done? It basically included a stretch of 2 weeks. Several morphological parameters were measured to check the effect of water stress. Measurements were taken randomly from different pods present in all groups. And the parameters are basically the leaves length, leaves width, number of leaves, internode length, and the outer leaves weight. There was different tests that were performed in order to check the effect of water stress. Number one is the steroids test, in which 1 ml of extract dissolved in 2 ml of chloroform. 2 ml of chloroform, then the addition of 1 ml concentrated sulfuric acid, which shows the appearance of reddish brown layers. It shows that there are steroids present in the basil plant. After it, tannins tests were performed, in which 1 ml extract taken from each group in separate test tubes was mixed with 1 ml of ferric chloride, which shows the appearance of brownish black color. After it, the saponins form test was performed, in which 1 ml of extract taken from all the 4 groups mixed with the 3 ml of distilled water, and that shows the bubbles formation upon shaking for 1 to 2 minutes. Flavonoids color test was performed by taking 1 ml extract from each group was shaked and taken in separate test tubes, and then the few drops of diluted NaOH were added to obtain yellow color. After it was treated with concentrated HCl so that the yellow color would fade away. After it, the alkaloids Meyer's test was performed. Firstly, the Wagner reagent was prepared by dissolving 2 grams of iodine and 6 grams of potassium iodide in 100 ml of distilled water, and 1 ml of extract from each group was dissolved in 1 ml of Wagner reagent to obtain the brownish reddish color, which would not appear in the basil case. Then there are results which shows the morphological parameters of the Ocimum Basilicum plant. Morphological features are basically include leaves length, mean, and their mean was 2.1 cm, 2.15 cm, and 2.1 cm. And it is according to the control group, daily group, and the day 1 group. And day 2 group, if we talk about the leaves length, that were 2.35 cm. Similarly, they are showing the leaves width, that is 0.925 cm, 0.9275 cm, and 0.95 cm. And in the day 2 group, it was 1.05 cm. Number of leaves are similarly 27, 28, and the 29, and then the 30. And there was also showing the powder leaves weight, which day 2 group has higher. Its control group has higher powder leaves weight, and the internode distance basically showed 5.5 cm, 4 cm, and the 5.5 cm. And in day 2 group, it is 5 cm. If we talk about the aroma, control group, daily group, and day 1 group have no strong aroma, and there was very strong aroma that was present in the day 2 group. Similarly, here positive sign indicates the presence, while the indicated the negative sign absence of the respective metabolites. If we talk about tannins, saponins, and the steroids, they have shown the positive results in all of the 4 groups, but the flavonoids and the alkaloids have shown the negative results, which mean there are no alkaloid and the no flavonoids present in the basil plant of all the 4 groups. And the appearance of black brownish color shows the presence of the tannins and the presence of saponins with the formation of the bubbles, and the presence of steroids with the formation of a reddish brown color layer. After it, there is conclusion and the final discussion. So basil plants grown under different water stress conditions exhibit different growth patterns, and all of the groups of basil plants have similar levels of tannin, saponin, and steroids, which have potential medicinal importance. Basil plants in all the groups lack flavonoids and alkaloids, which are typically associated with antioxidant or anti-inflammatory effects. And moreover, further research is needed to fully understand the implications of these results. Similarly, day 2 had the strongest aroma. The differences in aroma across the groups may indicate differences in the volatile compounds present in the basil plants, which could have implications for their flavor or medicinal